Okay, so this part gets into the microscopic and submicroscopic structures of muscle, which probably seem, not like less interesting than the stuff that you were doing in lab. It's the stuff that physiology depends on. So I'm going to try to take it all apart all the way down and then build it back up with you. Okay, so let's remind you of what you know. A whole muscle, right, is made of multiple fascicles. Each fascicle is wrapped in connective tissue. The whole muscle is wrapped in connective tissue. And then each individual fascicle is composed of multiple muscle fibers, which is what you're seeing here. And of course, the muscle fiber is wrapped in connective tissue as well. So what I want to do now is I want to go from the muscle fiber on down. Now, a muscle fiber is a muscle cell. So let's look at this figure and see what you can see inside this muscle cell. So things that you've already known is that each skeletal muscle fiber is multinucleated, 1 nucleus, 2 nuclei, 3 nuclei, okay? And it's striated, right? So the tissue looks striated. Remember, skeletal muscle looks striated. Pull that picture back up. So it looks striated. Right now, we're going to figure out why it looks striated. And then each muscle fiber, skeletal muscle fiber, is microscopically thin, but can be up to a foot long. So they're really, really long and skinny. Think like fishing line. So now what we're going to do is we're going to start breaking down and looking at all the structures inside the skeletal muscle fiber. Now, you spent a lot of time in 1610 learning the names of the organelles and intracellular structures. Unfortunately, some knucklehead decided to rename everything inside a skeletal muscle fiber. Sorry, they are still cell parts, but somebody wanted them to have the prefix sarco on almost all of them. So I will tell you what they are in a regular cell, and then I'll tell you what they're called in a skeletal muscle fiber. But it's not entirely different, and I'm sorry, I wouldn't make you learn these names, except these are the names that they use in physiology, and also going forward. So first thing is that let's acknowledge that this does not look like the cells that you looked at in 1610. This is a very strange looking cell. It is, though, just an animal cell plus primarily these big Vienna sausage looking things that allow it to contract. But let's look at the typical cell structures first, and then we'll add those in. Okay, so first off, every cell has a cell membrane on the surface. The cell membrane is usually referred to as the cell membrane or the plasma membrane, but not the cell wall, because that's not the same thing. But in muscle, they call it the sarcolemma. It is still a phospholipid bilayer. It's the same thing that you have learned in other cells, but it's called the sarcolemma. That's the cell membrane in skeletal muscle cells. Now, let's think about something for just a second. So every single muscle fiber, we said, had endomysium wrapped around the outside of it. That is connective tissue. That would be superficial to the sarcolemma. You're not seeing it here, but it would be like right here around. Okay, so that's not part of the individual cell. It's superficial to the individual cell. Okay, and then inside the individual cell or fiber, there is, of course, cytoplasm, also called intracellular fluid, but we have to learn a new name for it, which is sarcoplasm. And it's the cytoplasm in skeletal muscle cells. And it's got all this stuff in it. It's got, that's where the nucleus is going to be. That's where your mitochondria are going to be. That's where your endoplasmic reticulum is going to be. But in addition, you've got these big Vienna sausage looking things. I'll talk about them in a minute. But also there's a couple of important things in there that you can't see in a figure, but end up being functionally important. Inside skeletal muscle fibers, in the sarcoplasm of skeletal muscle fibers, the fiber stores glucose as glycogen. And glucose is, of course, used for cellular respiration. So it's basically got a little piggy bank of sugar that it can always use for its own cellular respiration. And then, of course, why so many mitochondria? Because this is a really energetically expensive tissue. And where does cellular respiration go on? In the mitochondria. One other thing is inside the sarcoplasm of a skeletal muscle fiber, you also store a little bit of oxygen on a protein not shown in this picture called myoglobin. Hopefully you remember that hemoglobin binds to and carries oxygen around in the bloodstream. Myoglobin binds to and stores little bits of oxygen inside the skeletal muscle fiber. So let me just remind you of something that hopefully will make sense to you. Basically what I've just told you is that the cell stores glucose and it stores oxygen and it's got boatloads of mitochondria. So this cell is always beautifully set up to, of course, make ATP, which is pretty damn important because this cell is, skeletal muscle fibers are the ones that keep you from getting hit by a bus when something comes at you. So the important thing about myoglobin inside the skeletal muscle fiber is that when oxygen binds to myoglobin, it makes the tissue red, just like when oxygen binds to hemoglobin, it makes the blood red. So that's the other reason that your steak is red. It's red because it's got so many capillaries in it, but it's also red because the oxygen is bound to myoglobin. So if you leave the steak out on the counter for a little while, it'll become less red as the oxygen dissipates off, or you can do it more quickly, of course, and you can cook it. Right? Okay, and that'll change the color as well. Okay, so inside, now let's address the elephant in the room. What the heck are these things right here? So you don't see those in normal cells. You don't see them in non-muscle cells, I should say. These big structures right here are big cable-like bundles of contractile proteins called myofibrils. So here's a myofibril, here's one, here's one, here's one. Okay, the more myofibrils you have inside a muscle cell or fiber, the stronger contraction it can do. Because what these do is they actually attach at the end to the cytoskeleton. And when you stimulate that muscle, they will actually pull, and they will pull on the ends of the muscle and shorten it. So this big thing is a myofibril. But do you start to see a repeating pattern in this myofibril? You got jagged line, light, dark, light, jagged line. So if you take all the stuff off so that you can see it really well, this is kind of what a myofibril looks like. Jagged line, light, this is not represented as dark, but it would be dark, a little bit lighter, dark, light, jagged line. So you have this repeating pattern in this myofibril. And once the pattern starts repeating from here to here, that is called a sarcomere. So I want to look at the sarcomere a little bit more closely. Okay, and you can see the sort of one jagged line to the next. This structure right there is a sarcomere. Okay, and the sarcomere is really where all the fizz happens. So this picture is really complicated, but let's simplify it just a little bit. So jagged line to jagged line, yes? That is flattened right here. Okay, this is three-dimensional. It makes it hard to do. Look at it 2D. Flatten right here to here. Okay, so the basis of skeletal muscle striations is this structure right here called a sarcomere. And get a different color. This structure right here called a sarcomere. And it is from Z line, also called Z disc, to Z disc. That is a sarcomere. And so before we get into how that's related to the physiology, I want to talk about how that's related to striations. So stick with me for just a second. So basically what you have is you have these two kinds of contractile proteins. You've got this one right here called myosin, which is represented right in the middle right here, and it's thicker. And you have this thinner one out at the edge called actin. And if I use a protein staining technique, because myosin is thicker, it will stain darker, and actin is thinner, it will stain lighter. So if I use protein staining, what I'm going to end up getting is the tissue is going to look darker wherever there's myosin and lighter wherever there's actin. So what we end up getting is we end up getting this striping pattern. So this part's going to be really dark because myosin is there. And then this part right here is going to be lighter because actin is there. And so we've got an A band, Okay, and then we've got a lighter band called an I band and another I band, and this would be the start of another A band over there. So this striping directly correlates to this whole light and dark striations that you actually see in a skeletal muscle fiber. It looks like that because of the actin-myosin repeating pattern in the sarcomere. And we're about to build a sarcomere for just a second. So let's make sure that we can do this. Just one, actually we'll do it right after we build our sarcomere. Okay, so basically, let's fill all of this stuff in real quick. So what we are going to have is we have the A band all the way down wherever there is myosin. We have an I band wherever there is no myosin. And there is a little period in the middle right here called the H zone. The H zone is where it's a little bit lighter than the darkest because I have myosin, but I don't have any actin overlapping it. So if you use a protein staining technique and you've got two proteins that are overlapping one another, this is going to be darker than the middle, okay? So that is called the H zone. So I band, whole thing here is A band from here to here, okay? And then this little middle part that's slightly lighter is the H zone. Okay, so now what I want to do is I want to build a sarcomere so that you can learn all the proteins, and I want to build it in your notes. So the basis of skeletal muscle striations and also the basis of skeletal muscle functionality is the sarcomere, and so we're going to make sure that we can build it and label things. So how is it the basis of skeletal muscle contraction? Because you have these two proteins that are going to overlap one another, and what's going to happen is they're going to reach up and they're going to pull. But let's build the proteins first, and then I'll show you how the pulling works. Okay, so in this figure that you've got over here from your textbook, what I'm going to do is kind of fill this one in so that the colors are similar. Now this one's way more complicated, but I'll put the parts that you need to know over on this side. Okay, so let's see what we can do. So the entire structure that you have right here, from here to here, the whole thing is of course a sarcomere, okay? But then we've got other things to label as well, and I'm going to try to do them in a similar color to the one that's on the notes or in the textbook. So this one that's in the middle right here with the little projections sticking up, that one right there is myosin. And those are the thick filaments, okay? And remember, they have little projections sticking up. And then the one that's primarily red over on the side here, these guys right here and here and here, these are actin, okay? The jagged lines at the edge, which I think are black over there, right here, over on that picture, are called Z lines, or this textbook calls them Z discs. Your phys textbook will call them Z line. It doesn't really matter which one they are. Okay, and then what else are we supposed to label? Okay, we'll do the rest in just a second. The whole thing is a sarcomere. Okay, we'll do the banding pattern in just a second. But what I want you to know is these little projections on the myosin that you can see right here, these little projections. Think of them as hands, and what they can do is they can reach up and grab onto the handles of the actin. So what's going to happen is the myosin right here is going to, when it's stimulated, reach up and grab the handles on the actin, and then it's going to pull. And as it pulls, what's going to happen is the Z lines are going to come closer together. So contraction actually happens at the sarcomere level. It does not happen at the whole muscle level without happening at the sarcomere level. So let me show you this figure so that you can see what's actually going to happen. So here's a relaxed sarcomere. a partially contracted sarcomere, and a completely contracted sarcomere. Notice that the H zone in the middle where there's no overlap is pretty big right here, and your I bands are pretty big as well. And then I contract it partially, and I actually get a little bitty H zone, right? Okay, and then I contract it completely, and I get a really completely disappeared H zone. Okay, so let's fill in just the last things on this diagram, and then okay, so let's go back to this. Oops, I spelled sarcomere wrong, knucklehead. The whole thing is a sarcomere, okay? So let's just fill in some places, right? So here to here, that's the H zone, okay? Let's fill in a different color, okay? Here to here, that's the A band, okay? One more highlighter color, and let's do purple. Okay, and then the I band is here to here, that's I band, okay? So I think we got everything. We got M, well, the M line is the one in the very, very middle. Make that one. That's just the stabilizing line that holds on to the whole damn thing, right? That's the M line. Okay, so we labeled M line, actin, myosin, Z line, H zone, A band, I band, sarcomere. We got it all in there. Okay, so the big picture understanding that's important to hear is when you stimulate a skeletal muscle fiber, some really interesting phys happens. You're not going to learn an anatomy yet. And then what's going to happen ultimately is the myosin will reach up and grab the actin, and then it will pull. And as it pulls, it changes the banding pattern. And what happens is the actin and the myosin filaments will slide past one another, and the Z lines come closer together. And the H zone can completely disappear, and the I band will get much shorter, but the A band really stays the same length because the A band is the length of the myosin, and everything gets pulled into the myosin. So this is called the sliding filament theory because I've got a myosin long skinny filament and an actin long skinny filament, and they actually slide past one another to cause contraction. Okay, so last little tidbit to put all of this back together with the big picture is if we go big to small and then small to big, then you can figure out how this is related to everything else. So let's talk, and we'll do this in class as well. So if we go whole muscle all the way down, and we can say, okay, how do we get down to the functional part? Well, a whole muscle, you can see, is wrapped in epimysium, right? And it contains a whole bunch of fascicles, yes? And an individual fascicle is wrapped in perimysium, and it contains a whole bunch of muscle fibers, right? An individual muscle fiber is wrapped in endomysium and contains a whole bunch of intracellular structures, among them myofibrils. And the myofibrils are the venous sausage-looking things that can be divided into sarcomeres. So if you take a myofibril and divide it down even further, you will see this repeating pattern from Z line to Z line, and that is a sarcomere. And if you take that and break that down a little further, what you can see is inside each sarcomere is a thick filament called myosin and a thin filament called actin. And that's how all of those things are related to one another. And we're going to practice this in class as well. Okay. Next video is just a short comparison to cardiac muscle and smooth muscle, and then this set of notes is done.